Hey, biology students. This is my first video outside this year. I'm doing this after school on Monday, so if you hear cars driving by or birds in the distance, sorry I'm squinting. I'm looking at the sun here, but it's too nice to be inside, so I'm out here working. So I want to talk to you about this genetic disorders disease project that I'm going to ask you to work on. So the purpose of this, and I know this is probably a little bit small, I'll make it a little bigger. In fact, let me actually shrink myself down here. All right, so I'll shrink myself down here a little bit. So really, the purpose of this is to investigate multiple genetic disorders that are somewhat common. Even though when I say the word common, doesn't mean that you're going to meet somebody every day that has one of these, um, but there are ones that are relatively more common. Allow you to apply some previous, some already skills that you've already learned about, some terminology, and also learn some new terminology. And I'll sprinkle some of that in here today. So you're watching this slide now. I have a list, and so actually let me just jump to the list. So I have a whole list here of 14 different genetic disorders. Now, before you get too worried, I'm going to ask you to do at least 6 of these. And I've already done one exemplar, and again, I'll talk about that in just a moment. These are no certain order. I should have alphabetized them perhaps, but I didn't. I just kind of brainstormed my ideas. Um, and so some of these are going to be very big changes. Some of them reduce the life expectancy of the human quite a lot. And these are all human genetic disorders. But what I'm going to ask you to do is to do at least 6 of these different disorders. Hopefully, by the time we get to the end, that in a class, they've all been done. That's kind of my goal. So we can look at other people's presentations and kind of learn about these. These are ones that I've picked that we will use as we move on to our genetics unit. So if I'm talking about Huntington's disease or Tay-Sachs or sickle cell or deuteranopia, which is a tough one to say, but a lot of you know about that. It has to do with color blindness, actually. Hemophilia is a blood disorder. Cystic fibrosis is a problem with the lungs. We actually had a student a few years ago that did succumb of her symptoms of that. She had already had a lung transplant. Those lungs were sort of rejected, and she did pass away from her cystic fibrosis. Turner syndrome is when it's on the X chromosomes. We actually saw that when we were doing karyotypes, as well as Klinefelters. Achondroplasia, that has to do with we'll say your bones and whether they grow or what they don't grow. Marfan syndrome is another one that has to do with connective tissue and bones. Albinism, most of you have probably heard of albino organisms, and so what those are. And I'll go in. And Stargardt's disease is a new one that I've just learned about that is relatively rare, but it is a gene that is in the Danville area. So thought I'd throw that in there too. Now, so what I want you to do, or so there are criteria that I want you to examine. So when I say criteria, there's just different things to look up. Now, there's a lot of this is a lot of stuff here, and I would recommend that you view this pretty large. And I'm not going to necessarily read every single little detail to you, but so criteria 1 is the most common symptom or symptoms. So what stands out as to the problem or the issue with that person. Second criteria, which chromosome is it located? So you're going to identify the chromosome. Is it an autosomal? And in fact, I should have probably moved. Is it autosomal dominant or autosomal recessive, sex linked? We talked about autosomal. That's when it's on chromosome 1 through 22. If it is sex linked, then it's on the X or the Y chromosome. I actually do not have any diseases that are on the Y chromosome. There are some things that are linked there, but there are only about 300 genes on the Y chromosome. So there aren't a lot of diseases that are associated with that. Also, the location on the chromosome. So when I say the location on the chromosome, for instance, you're going to see this as maybe this is a disorder, and so it's 12p23.3. What that means is 12 is the 12th chromosome. So there's which chromosome it's attached to. P is the P arm, and then there are going to be bands along that chromosome, and this is band 23.3. You could think of it as being a measurement as measured from the centromere. So the centromere would be kind of 0 as you move away from the centromere, whether you're moving away in the P arm that's up or the Q arm that's down, that will increase the number. That's sort of like the address of the gene. If you find yours is XQ39, then that's telling you it's on the X chromosome, the Q arm, and it's at location 39.0. So again, that's sort of the location that you're looking for. We talk about dominant recessive. You'll find that those are terms. Dominant means you only have to get it from one parent. Recessive means you'd have to get it from both parents to have that disorder. The type of mutation that we are. So this can be tricky to find for some of these because um, you will find if it's a point mutation or chromosomal alteration. Chromosomal alteration, and there are very few of those actually on my list, maybe only one or two. So if you can find this as to what's the mutation, is it a substitution? Is it a deletion? Is it an insertion? You may find some information about that. So and I'm going to sit down and do all these as well, so I'll know what you should be actually be finding. Um, this may be the trickiest of them to find. Is what kind of mutation is it actually? Is it a point mutation, a single letter, or a small number of letters? Or is it a big change in inversion? And the thing is with these is it's a mutation of that gene, but it could be any one of many. So the exemplar that I found, they actually said there are three different variants, three different mutations that can actually occur. Sometimes it's a stop mutation, it's stopped too early. Sometimes it's a mutation that causes the protein to have the wrong shape. Third criteria, name the protein that's affected. Now, the names of the proteins are going to be really weird, and I'll show you that in a moment with the exemplar that I have actually done. And also, if you can find what's the length of the normal protein, what's the length of that normal amino acid chain? Is it 334? Is it 225? Is it 1018 amino acids? How big is that normal amino acid? It's just something that we've talked about, like amino acids, so it's kind of applying that information. Criteria four is the prevalence of the disease or disorder. Most often with this, you'll find it as a percentage of the population. You might find it as, so I said you might find a percentage of the population. You might actually find it as a ratio. So, for example, you might find it happens in 1 in every 10,000 individuals, or it might say 1 in every 10,000 births. So that just means if you meet 10,000 people, chances are one of those people has the disorder. It's sort of a probability, is the ratio that we have here. And then if you find it runs in any ethnic groups, if it runs more in boys or girls, so any information you give me about prevalence, how often it happens. So in ethnic groups, for instance, sickle cell anemia, you're going to find that happens in really high in one ethnic group more than in others. I don't want to give anything away, but you'll often find that. And now when you Google search these, it might just be going, and I'm fine with using the Wikipedia page. The Wikipedia page for these are not lies. They are pretty factual. That is fine. And they do a nice job of showing you which chromosome. You can click on the chromosome. You're also going to find that there are going to be some words that you don't know what they mean. That's okay. If you're in person, ask me. I can help you decipher. Sometimes it's going to be words that I don't know. But we're really looking at the big picture here of these disorders. And then criteria 5, how does the disease, if any, affect the quality of that person's life or their life expectancy? Okay, so how did I approach? So let me give you this exemplar. So an exemplar, really what that means, if you hear that word, it means here's a model, a really good model of what I would consider to be a 4-point entry. So notice how I've done my criteria. What I actually did is that I listed, so here's my symptom, here's the gene and chromosome, that was criteria 2. The protein affected, there was 3. Prevalence was 4, and quality of life was 5. So notice how I did that. Notice I don't have any fancy pictures here. All of my information fit on one slide. However, if you find that you need to put it on 2 slides, that's fine. I would try to make it so that your font size is never smaller. I think my smallest was about 20-point font. I'd be careful going any smaller than that. Notice I don't really have complete sentences here. I have very much sentence fragments. That's what I'd like to see. Now, there are a couple sentences, but be careful with complete sentences. We just want fragments here. This exemplar is for a gene that's called the PTC gene. This is a gene this isn't necessarily a disorder, but it's a characteristic that lets the folks that have this, it builds a taste bud on your tongue that helps you taste the chemical PTC. It's a big, long name. It's not that important, but it's a really bitter taste. And it's in a lot of vegetables. It's actually in the family Brassica, which are your cabbages, Brussels sprouts, things like that. Excuse me. If you when you taste Brussels sprouts or broccoli and it just tastes like terribly bitter to you, you might have an overactive one of those senses of those sensors. Now, as it turns out, 70% of the people have this gene. So, and it's not a mutation, it's actually normal to be able to taste it. The mutation is not having that taste. So if you can, when you taste broccoli and those things, and really talking like the raw food, if it's not bitter to you, and I actually have a strip of paper here in school that will test whether you have that or not. It is a bitter taste, and I do taste it. I do taste the bitterness. So the symptom: oversensitivity to bitter tastes. This explains why some people hate broccoli and cabbage, and a lot of kids they don't like the taste of that because they are sensitive to that. The gene that it's located, it is actually on the seventh chromosome. It is an autosomal dominant. So I didn't actually find it was autosomal. What I found is once I knew that it was on chromosome number 7, that I knew that was autosomal because it wasn't the X or Y chromosome. And dominant I did find, so that means you only have to get it from one parent to actually have the taste. The location, that location is on the Q arm at band number 34. So that's its address. Again, that's its address. And now the mutation, there were actually 3 different variants that I actually found, but it said most commonly it was actually a premature stop. And that does actually make sense because you have to remember that what you have on your taste buds are like these little, it's like a little receptor. And so what's supposed to happen is the smell is supposed to dock into that receptor. And so think of it like that receptor I talked about with coronavirus. And so if you don't have that receptor, you can't taste the bitter flavor. All flavors and tastes are actually based on a molecule. So your tongue has these taste buds that little molecules will attach to. It's also how you smell. And that's then what sends that message to your brain. It says, hey, this is bitter, this is sweet, this is sour. Or if you're smelling things, it's also those chemicals that you smell that fit into different chemicals in your, so they'll call the olfactory bulb that's up here in your nose. So I just said it was a premature stop. And again, you may or may not find that information about yours. The protein. So here's the protein. It was called the TAS2R38. What the heck? Where does that come from? Well, I think what TAS is the taste, has to do with a taste, is probably what this is. And then 2R38 probably has something to do with the protein, but that's the name of it. And often proteins are given this weird name. It was called a taste receptor. I did find that it's 1,002 letters, so it's 334 bases long, or 334 amino acids long, from met all the way to where it stops. And really the function is as a receptor to taste, to sense that PTC molecule, which is a very bitter taste. Prevalence, actually 30% of people do not have this. So it's one of those things that this isn't necessarily a disease, but it is a characteristic. It is a gene. So that's why I wanted to use this as an example. Interestingly enough, almost all Native Americans can taste that bitter. They find that only 2% of people that are actually non-native to North America cannot taste the PTC. So, and that sometimes makes sense. If you're from a small group, so originally the people who moved to North America through the land bridge up through, across from Siberia over to Alaska and then moved down, probably everybody in that group could taste that. And so the genes have gone unchanged. So only a small number of people may really originally inhabited North America. And if your ancestors could taste it, you can taste it, unless there's some sort of a mutation. And that's probably why 2% can't. There's just a small number that have a mutation that you can't taste it. Quality of life. Now, I don't know that there's a whole lot of quality of life here that's changing, other than maybe if you're a non-taster, you can enjoy broccoli a little bit better. You have a healthier, and Brussels sprouts. But here's what I thought was interesting. They thought the gene actually evolved for this taste bud to help us with some toxins. There are things in nature that have a really bitter taste that are toxic. So maybe that's where this actually evolves from. Pretty much all of our genes that we have, they're there for a reason. It's because Mother Nature intended that, and it helped us survive longer. And so those things got passed on. So that's my exemplar slide. How am I going to grade your slides? Well, so if you do 6 of these, 6 of the projects, 6 of the required 6 different disorders, each one's worth 4 points. There's my hand. So how do you get a 4? All information is accurate and free of errors. All requested criteria is added to the slide. So all 5 of those criteria, you've addressed that to the best of your ability. The slide is organized. When I say organized, I'd like you to really use mine as a model. There's nothing wrong with you actually duplicating my slide. And if you don't know how to duplicate a slide, you click on the slide, you go up. Sorry, that's a loud truck. And you hit duplicate. It will duplicate that slide, and you could just delete everything and just leave, use the bullet points. So there is nothing wrong with that whatsoever. In fact, I'm not going to add any more slides. What you actually do is you add slides to the end of this presentation, however many you think you need to add. Maybe some of the disorders you find lots of info and you want to do 2 slides for that. That's okay. Remember, 20 point is about the smallest I want you to make your font size. Really don't think the investigation should take that long. Each one should be done in a pretty short period of time. There's not a lot of really deep investigating. If you're working on one, you're like, I cannot find anything on this. What should you do? You should find a different one. You're being asked to do 6 of the 14. So there are definitely some there. If you're having difficulty, come to Mr. Acker and say, Mr. Acker, do you have a recommendation? Because I'm batting, not doing very well here. I'll point you in the right direction. Okay, but I think there's going to be information for all of these things. All right. When you're done, then just please share it with me, or don't share it. Excuse me. You'll go back into Power School. That was wrong. You go back into Schoology, and you will submit your presentation. Um, if you find that you say, hey, can I do a 7th one, Mr. Acker? Could I get more points if I did a 7th one? Yep. If you want to do all 14, then 14 times 4 is 56. You can earn up to 56 points for this. So, yes, you could do extra if you wanted to, and I will reward that extra work. We won't call it extra credit. We'll call it extra. It sort of is extra credit, but it's not like out of 0 points. So, all right, hopefully that's helpful. Hopefully my exemplar is helpful. I know sometimes that's helpful to know, like, what should my slide look like when I'm done. If you have difficulty understanding any of your sources, please let me know, and I will help you out decipher some of the terminology. All right, thanks so much. Have at it. Ready, set, TAC.